Our first speaker is Dr. Irilenia Nobeli. She's a lecturer in Bioinformatics here at Birkbeck and Program Director for our MSc Bioinformatics. And she's going to talk about AI, what it is, what it isn't, and why should biologists care. Over to you, Irilenia.
I'm hoping, I switched on the mic, right? I think, no, switch that off, that's off. Right, so welcome everybody and welcome at home as well. I think I'll start with a small confession. I'm not an expert in AI, and probably that's not a very good start, you might think. But actually, I am the right person, I think, to give an introduction to AI, because I think if you were to get an expert in AI, you'd probably be looking at about 30 slides on linear algebra. And probably that's not what you want to do on a Tuesday evening or any other evening. So it's good you got me instead. So I know what is your next thought, and your next thought is, if she's not an expert, did she get an AI chatbot to write a presentation for her? And I have to say, I tried. I did try. I said, what should I include in a presentation like this with this title? And ChatGPT was actually quite helpful. It gave me a very long, it's not just this, it gave me a very long list of things I had to include and not include. But it was very generic stuff, and I realized, I'm going to spend a lot of hours talking to this chatbot, trying to get the presentation going, when I can actually do it myself in the same time. And so I decided to do it myself, and another reason why I decided to do it myself is I actually quite like writing lectures and giving presentations. So it's not like I needed the ChatGPT to do it for me. If AI wants to help me, it can come and clean my bathroom, actually, or pick up the kids' stuff off the floor, but fat chance of that. So this is a human-made presentation. It will probably include human errors. I hope not too many, but there may be some. Right. So AI, what it is. So one thing I want to make clear is I think there is no controversy on the artificial bit. Artificial means non-biological entities, so it's not nature-made. So we're talking about machines here. And the second bit is that when we're talking about intelligence, that's where the problem comes, because that's a very hard question to answer. What is an intelligent machine? And we can say perhaps intelligent machines are machines that do intelligent things. They show behavior like us. So we are intelligent because we have rather large brains for the size of our bodies, and we can actually display non-trivial behaviors. We can do things. A machine may be intelligent if we can do things that are useful to us or entertaining to us, and so on. But all of these definitions have various issues with them. If you think about it, they do actually have many issues. Talking to a friend recently, he managed to convince me that his fridge was intelligent because it was doing something very useful that no human could do. And so I'm moving away from that definition, and I want to narrow it down today to a definition of the following. Something is intelligent if it can acquire knowledge, it can learn, and then apply that knowledge to solve problems and achieve objectives. And these objectives don't have to come from the machine itself. It can be us that give the objectives, but the machine is using the knowledge it acquired to actually achieve the goals. Right. So what it is not. First of all, it is not many things, but it is not recent. And I say that because if you know Blaise Pascal, he created arithmetic machines back in the 17th century, and many people have been trying for hundreds of years to create all sorts of machines that are intelligent. But we probably associate more the beginnings of machine intelligence with these two people, Alan Turing and John von Neumann, both polymaths, both extremely intelligent people, who really put the foundations of what we think now of as information processing in computers, basically. And without them, I think we wouldn't have got to where we are today. But artificial intelligence, really as a term, was coined in 1956 in what is now known as the Dartmouth Conference, and that was organized by Rochester, McCarthy, Minsky, and Shannon. And it is thanks to these people that we use the term artificial intelligence today. Artificial intelligence is also not what we generally call general intelligence. And you may have heard the term artificial general intelligence, or AGI. And it is not AGI because currently AI is not anywhere near being the type of intelligence that can solve any type of problem, that can apply its knowledge to various domains, and that it doesn't care about what it's been trained on. So we're not quite there yet, but you would be a fool to think that we will never get there. I don't think anybody thinks that. And also, we don't really know how close we are to that point, which is another problem. And I wouldn't put any bets at this point, but it does not yet replicate human cognition. So unless you lived under a rock for the last 2 years or so, you may have heard not only about artificial intelligence, but also the terms machine learning, deep learning, artificial neural networks, and so on. And I'll try to talk about all of these so that Caroline doesn't have to do that when it comes to her talk. So what's machine learning? So essentially, when we program computers, there are two ways of doing it. There's the traditional way and the machine learning way. In the traditional way, we have a problem, and we already know the answer to that problem, and we know it so well that we can give really accurate instructions to a computer to solve the problem. We can write down the instructions, and the computer goes off and does it. And if we get the logic right, the computer will not only do what we ask it to do, but it will actually do what we want it to do. If we get the logic wrong, it will do what we're asking it to do, but it will not do what we want it to do because we got the logic wrong. But essentially, we know the solution. We can solve limited problems because it's very specific what we're trying to solve. And we can step through the program, and that's important. We can step through the program, understand how it works, and also pick up any mistakes in the logic that way. Now, machine learning, on the other hand, is a non-traditional way of doing things where we either don't know the solution to the problem, or we know a solution that's very inefficient, or we know a solution that we don't want to tell the computer because we wanted to find a better solution or something like that. So for various reasons, we don't want to tell the computer what to do. But what we do have is a lot of data. We say, okay, here's the training data. We feed it to the computer. The computer comes up with some model. There's a model, that box here. And then once it has built a model, it has found patterns in the data. That's what it's trying to do. It finds patterns in the data. We feed it new data, and it can make predictions out of it. Okay? So there's a reason why I put the model as a black box, because that's exactly what it is most of the time. In machine learning, the model is often a black box. You can't open it and see what's going on. We can't really, it's very hard to find out what the computer is thinking it's doing there. And because of that, we can't easily also play around and fix things with it. Right. So that's machine learning. What are artificial neural networks? So when people started thinking about how do we build intelligent machines, they said, well, we've got quite a lot of examples of intelligent machines all around us. Obviously, we've got also examples of non-intelligent machines around us. But if you look at humans, we're supposedly intelligent. And if we look at our brain and understand how our brain is made, perhaps we can make machines that look like our brain. And if we make machines that look like our brain, then the machines will think like us. Yeah, that was the whole thinking for at least 50 years. So what does our brain look like? We have a lot of neurons. And when I say a lot, we've got something like of the order of 100 billion. Now it's probably more like 86 billion, but that's the order we're talking about. And the number of connections between these neurons is a lot larger. We have 100 trillion connections. Okay? So people thought, okay, let's try and build something artificial that's made up of something that looks like neurons and has lots of connections. Then we can solve problems. And so they started off with this simple idea. What does a neuron look like? It's something that takes inputs from the environment or wherever and produces outputs. Here, it's a single output, but it could produce multiple outputs. Okay, so that's an artificial neuron. So what do we do then? We make lots and lots of copies of that. And we will build a network. But before that, I just want to say that another important realization is that not all inputs should count the same way, right? If you're thinking about it and, your mom is calling you and then your gaming mate is calling you, who are you going to pay attention to, right? So there's a weight to the calls, and it's a similar weight here. Some inputs are much stronger than others. Okay? And also what happens is in order to produce the output, you have to do something to the inputs. So you need a function in here that does something to the inputs. It turns out, again, after a lot of trial and error, that some very simple functions are the best. And the simple function here is you do a sum of the weighted inputs and produce the output as the sum. And if the sum turns out to be negative, you produce 0. So that's what this plot is showing. And this has a very fancy name called ReLU, and it's the most common used function in artificial neural networks. Right. So a network is made up of all these artificial neurons which form layers. So everything vertical here is a layer. Yeah. And you have an input layer where the inputs are coming from outside, basically. And they're fed into the inside layers, the hidden layers. And then there is something that receives the outputs of the last layer and produces an output, and that's the output layer. And all these hidden layers are very much interconnected, so everything is connected to everything before it and everything after it. So the layer before it and the layer after it. Okay, so there are loads of connections. And when we started off building artificial neural networks in computers, we couldn't efficiently train them, so we couldn't use many hidden layers. The whole thing was a bit of a disaster. So eventually we got better algorithms and better computers and managed to increase the number of hidden layers to a very large number. Now it turns out that that's what you needed in order to create something with intelligent behavior. You needed a lot of hidden layers. And a neural network that has a lot of hidden layers is called a deep learning, basically. That's the deep is the depth of the hidden layers. Okay. So this may be a little bit of an oversimplification of what goes on in computer science. So if there are computer scientists, pardon me, but I'm trying to get the point across, okay? Okay, so where's the biology? I talked a lot about AI, but I didn't say anything about biology. So many problems in biology are actually problems of finding patterns. And we as a human species are excellent at finding patterns, actually. We are very, very good. We can look at pictures and find patterns and so on. But we can only do it with simple things. We can't do it when we have billions of points of data. Okay? And this is where we need artificial neural networks, because they are very good at finding patterns when there's a lot of data. In biology, we just have a lot of data currently. So this is where the biology comes in. Now, I'm going to talk about a single application of AI to biology, and then Caroline will talk about her own research, which is much more focused. And this application, obviously, it has nothing to do with me. This is people a lot more clever than me that came up with it. But the application I'm going to talk about is the protein folding problem, and that's a 50-year-old riddle. In order to say what protein folding is, I have to say what proteins are, because just in case there are people who haven't done biology yet. So a protein is basically a polymer. It's made up of building blocks, like Legos are. So it's a chain of building blocks, and the building blocks are called amino acids. And every amino acid is represented by a letter. So when you look at the sequence of a protein, you're looking at basically at an ordered sequence of letters from a specified alphabet. So the alphabet is 20 or 21 amino acids long, it doesn't matter. But it will look like this. Every letter is an amino acid, okay? And the proteins are what does everything in our cells. Basically, nothing would work in your body if you didn't have proteins. They do everything, pretty much everything. But in order to do something, when they're created, they come out of these massive factories known as ribosomes, and they are long and unstructured chains. They're basically long chains of amino acids. And in order to do something, they have to fold. They have to create a three-dimensional fold. Okay? And this is an example of a fold. I mean, it's a diagram of a fold, but they look really beautiful, these three-dimensional arrangements of amino acids. And what's the protein folding problem? Well, this started with this guy here, Christian Amundsen, who, when he accepted the Nobel Prize in Chemistry in 1972, in his speech, he said, oh, if you have the amino acid sequence of protein, that's sufficient. You can get the 3D structure of it. And rather unhelpfully, he didn't tell us how to do it. He just said it should be possible, because he had done experiments that showed that it doesn't matter what your starting point is. As long as you have the same sequence, you will always get the same structure at the end. So the sequence is enough. Yeah. So he didn't tell us how to do it. So he left us with 50 years of struggle to solve that problem. Now you may think, okay, it's not that difficult. Actually, we can just try all possible conformations in 3D and we'll find the one that's right. And here comes Mr. Leventhal, Cyrus Leventhal, around the same time as Amundsen. He proposed the Leventhal paradox. He said, well, if you have an average length protein, about 150 amino acids or so, it would take longer than the age of the universe to try out all the possible conformations. Because the possible conformations are something like 10 to 140 or something for a normal size protein. That's 1 with 140 zeros after it. Yeah, that's very long. So if you have that many conformations, it will take you longer than the age of the universe. And that cannot be possible because when the proteins come out of the ribosome, they fold within, milliseconds, sometimes seconds, sometimes milliseconds. So they don't take the age of the universe to fold. So they cannot be possibly trying out all possible conformations. In fact, it turns out that this is the case because actually once you find the path to go down to what we call the global M energy minimum, this structure, you're going down very fast, that path. You don't need to search the whole space. And the same thing when we do that, when the proteins do that, they do it like this. When we try to find the solutions, we should also not look around the whole space. Right. So before AI came along trying to suggest solutions to this problem, there were essentially two approaches. Again, I'm oversimplifying things, but there were essentially two ways of going from the sequence to the 3D structure. What were the ways? One is what we call the ab initio method. Ab initio basically means from first principles, right? And this is a beloved method of physicists because they like first principles. So they say, if we understand what goes on between atoms, you can actually predict what each atom, how each atom will interact with every other atom, then you can fold the whole thing. Yeah, that's a simplified sort of presentation of the ab initio method. So you need to understand how atoms interact, and you need to model the interaction sufficiently in a computer in order to do it. Yeah? And none of these two things is actually true. We can't do really well either of these things, and this is why the ab initio methods don't work as well as we would like them to. So instead, we cheat and use what is known as the template method. In the template method, we say, so the proteins fold, but they belong to families because of evolution, because of things like gene duplication and so on. Some proteins have very similar sequences and they tend to have the same fold. Yeah? The details differ, but the overall structure is the same. And because they belong to families, if you know one member of a family and you know what the fold looks like, then you're okay because you can use that fold as a template to actually fold the new sequence you have. So that's really good news for us, and it's basically what solved a lot of protein folding problems in the past, having a template. Okay? So you tweak the details. Now the problem with that is that some families do not have any structures known, so we can't rely on a template. And the other problem with that is that you still need to tweak the details, and the devil is in the details often. So sometimes you produce a structure that is not accurate enough to do anything with it. It looks overall good, but the details are wrong. And so that's not very helpful. Right. Why do we care about predicting structures? Maybe it's a time to pause a little bit and think about this. So actually, what I didn't tell you is that the structure and the function are very intimately linked, or I hinted at this, but perhaps I didn't emphasize it enough. So if you have a structure, you can most likely predict the overall function of the protein if you're lucky. So it helps us to predict and understand function. It helps us to engineer new proteins. We can actually tweak things in the structure and produce new functions. And the most important thing for us possibly is that a lot of pathogens have proteins that attack our body and we want to stop them. And in order to stop them, we have to create inhibitors, drugs. In order to create those drugs, we need to understand how they interact with the proteins, so we need the structure. There's also a lot of talk about misfolding of proteins that lead to disease, like Alzheimer's, right? If a protein doesn't fold well, it can cause problems. So we need to understand how to reverse this misfolding. And the other problem we have is that we know millions and millions of sequences, so we know probably about 200 million of sequences right now, but we only know a few thousands of structures because it takes a PhD's worth of work to solve a structure, or we used to at least. and quite a lot of money. So we only have very few structures. So we want to close that gap. And this is where AI can help us. So I'll take you back in time a little bit. Do you remember what you were doing on November the 30th, 2020? Probably not. But if you opened any newspaper on the day, you would have got this. So you have got something like DeepMind AI cracks 50-year-old problem, or one of biology's biggest mysteries largely solved by AI. And I even put Daily Mail there because I thought they probably didn't realize that the CEO of DeepMind, Demis Hassabis, is actually the son of 2 immigrants. So I thought it was quite hilarious that they were celebrating too. Right. So how did we get there? How did we get to AlphaFold? In March 2016, DeepMind, that London-based company that I just mentioned, built a program called AlphaGo and challenged the Go champion at the time, Lee Sedol, the world champion at the time, to a 5-game match. And he won 4 of the 5 games. The computer won 4 of the 5 games. It was the first time that this happened. Go is an ancient Chinese game. It has extremely simple rules, but it's extremely difficult to write a program that will beat a human in it, because it's a very strategic game. It has 10 to the 700 combinations. Compare that to 10 to about 100 chess possible games. Yeah, Go has 10 to the 700 possible games. So it was quite an achievement. And on the way back, the team that built AlphaGo, on the way back from Seoul, already on the plane, they were planning what their next move was. And the next move was, we're not going to play games anymore. We're going to solve a serious science problem, because we've got now computers that can do this and software that can do this. And so they came up with AlphaFold. And AlphaFold was a computer program that basically was trying to predict structures from sequences, and did extremely well. They took part in a competition in December 2018. That competition is very famous in structural biology. It's known as CASP. And basically what happens is you get loads of sequences and you have to predict the structures. And the organizers of CASP know what the structures are, but they haven't released them to the public. So it's a very fair competition. Nobody has the solutions. Everybody's trying their software on exactly the same proteins. So it's a really, really nice test of how well you're doing without knowing the answers. AlphaFold came from nowhere in structural biology and came fast right to the top, and was a massive difference from the second thing. But what it didn't do is it didn't reach experimental accuracy. And structural biologists were, yeah, you're doing well, but you're not doing that well, are you? We still need X-ray crystallography to solve the protein structures. And so 2 years later, DeepMind said, well, hold my beer. November 2020, AlphaFold 2 was out, and it produced the most amazing results we have ever seen in structural biology in terms of structure prediction. So, what this plot is showing is basically the error you get from the top 15 pieces of software that took part in this competition versus AlphaFold. And they're all about at 3 angstroms, which is extremely good, yeah, but not good enough. And AlphaFold is at the experimental level of accuracy at 1 angstrom. This is an average across 80, 90 protein domains. So this was a massive success for AlphaFold. And I want to actually show you. This is one example. This is a bacterial protein from the same competition. This is the experimentally derived structure in gold. And in cyan is the prediction from AlphaFold. And it's just almost perfect. I mean, it's a well-chosen example, but many of the examples were that good. And the interesting thing is that the original protein has this zinc atom there, so it's a metal, and it functions only if the zinc is there. And AlphaFold predicts also really nicely the binding site of the zinc. And now that's a bit weird, and it tells you that AlphaFold is very good, but doesn't know any physics. And it doesn't know any physics because it doesn't see the zinc atom. So if it doesn't see the zinc atom, it shouldn't really arrange these amino acids and histidines around it like that. It really should arrange them in a different way. But what it has learned is that there are many zinc binding sites in the proteins it trained on. So it trained on a lot of structures that we already knew. And it knows what it should look like. Yeah. And that's a question. Do we want really a program that doesn't understand physics, but knows what you might want to have there? Or do we want a program that understands physics? That's a question for another day. So we then had an explosion of structure predictions once AlphaFold became available. They basically predicted 350,000 protein structures covering the whole proteomes of humans, so human proteins, and another 20 model organisms. These are organisms that biologists are very interested in. Yeah, so that was within a year, or less than a year actually, from the competition. And 1 year later, they uploaded 200 million structures. So now we are on par with our knowledge of sequence. The knowledge we have about structures is the same as the knowledge we have about structures, more or less. Sequences and structures, sorry. But I don't want to brush under the carpet the fact that AlphaFold is not always that wonderful, right? So there are problems, and here's a problem. There's a structure, and if you know anything about structural biology, you know that the blue bits look quite convincingly as structures, but the things that look like spaghetti doesn't look like what we would expect a protein to fold like, okay? And this is an AlphaFold prediction of a kinase in humans. And so we don't actually know why it fails to predict these regions. The people who wrote the software believe these are truly disordered regions. The protein doesn't fold. Not by itself anyway. It may need the presence of other things. And they may be right partially. I think a more parsimonious explanation may be that actually this is a machine learning software that cannot generalize well in cases where it doesn't know. It doesn't have a good example to learn from. So these may be regions that it has never seen in the protein database. So in the basically the database where it takes the structures from to train. So if it hasn't seen them, it doesn't know how to fold them. So that could be a problem. But we are in a situation where we have a structural abundance, right? We have many, many structures, and we can do a lot of things with them. One of the things we can do with them, and Caroline will show other things we can do with them. One of the things is that we can really solve massive structures that are made up of many proteins. So this is the nuclear pole complex. This is a massive complex of about 1,000 proteins that acts like a gatekeeper of the eukaryotic nucleus, right? And each one of these proteins, we needed to know the structure of in order to build the complex. Okay, but we didn't really know them. Now we know about 2/3 of them, thanks to AlphaFold. So we can build a very convincing reconstruction of it. Another thing we can do is to look back in evolution and try and find new families of proteins. So, for example, we have here AlphaFold 2 was used actually by a group at UCL that we are very close to, Christina Orengo's group. So they predicted structures for 21 model organisms using AlphaFold, and then compare this to their database of families, and found that there are quite a lot of new families they hadn't thought of. And it's a particularly lovely one. It's a heart-shaped structure that is entirely new fold. So how does it work? I'm going to just spend the last few minutes telling you a few things about how it works. This is where normally people come to. They take this, they take this figure from John Pearl's paper. That's the AlphaFold paper. And then try to walk you through it. And I think that's a bit of a horror show. So I'm going to do a dummy version of this. But I'll start with the two basic principles that AlphaFold 2 is using, and then I'll show you my dummy version of the same figure. So the two basic principles that AlphaFold was based on was a very old idea known as coevolution, and a very new idea that I'll talk about in a second. So the very old idea relies on the fact I told you before. The protein sequences have relatives. They are like parts of a family. They're separated into families. And the families have sequences that look alike. So if you look at every line, you will see the sequences are very similar. But there are differences, yeah? So here the R is replaced by K and so on. And if you look at the separate columns, you will see that sometimes when a letter changes here, another letter somewhere else in the sequence tends to change too. And that's something to do with evolution, trying to keep the 3D interactions we see in the 3D structure intact, trying not to break them. Yeah? And you get this effect, you get this covariation going on between columns. And structure prediction programs often relied on this they use that property in order to understand which residues are actually interacting are close in distance in a 3D structure. So it's a trick, basically, okay? They use that, and they also use something much more modern, which is known as transformers. Transformers is a type of neural network. It's basically what ChatGPT is a transformer. And transformers rely on this thing called attention. In fact, this is from a paper that has the title, "Attention is all you need." And that's a reasonably new idea that was published in 2017. And what do they mean by attention? These transformers were initially used for machine translation, so translating text from one language to another. In order to do successful translation, when you take a sentence, you cannot translate word by word. You're going to have to look around the context of the words in order to get the things right. So in these two sentences, for example, the word banks has two entirely different meanings in the top sentence and the second sentence. But in order to get the translation right, you have to find which word in the sentence you need to pay attention to when you translate in banks. So in the first case, cash might be a good one. The second case, the river might be a good one. But also others could be burst, for example, or withdrawal. Okay, so this idea of paying attention to a specific part in a sequence turns out to be an extremely useful property to solve a lot of problems, not just translation. And this is what they're using also in AlphaFold. And in one way they're using it in several ways, but in one way they're using it is that they're paying attention not to a single amino acid. So if you think this is an amino acid, and that's an amino acid, and that's an amino acid in space, they're looking at amino acids in triangles, in sets of 3. So they're paying attention to one amino acid and two neighbors, if you want. And the reason they're selecting these triangles is because of the triangle inequality. So if you know this distance, or you know more or less that I and J is close to each other, and J and K are close to each other, then automatically you have a distance constraint for K and I. That's pure geometry. Okay? So that was a very clever trick. Nobody had thought of that before to apply it in this type of predictions, and works really, really well. So there's my for dummies version. You start with a sequence. You supply a multiple sequence alignment because you need that information of coevolution that I talked to you about. You give it a rough guess of where the residues lie. And you also use templates if you have them. If you have structures from similar sequences, you use those. And then you have this massive black box where it's called evoformer. So this is where the artificial neural network, this is where the transformers sit, right? There's a transformer for this, for the pre-representation, and a transformer for the MSA. And a clever trick is that these two things exchange information in here. So they're trying iteratively to improve this pair representation and the multiple sequence alignment by going around the neural network again and again and again and again, about 48 times, I think. Then out comes an improved MSA, out comes an improved guess of the residues in contact, and then they have yet another neural network, another black box, that will take those two and produce a 3D structure. And that 3D structure initially might not be a perfect match. It might not be a great guess. So then they feed that back into the network and going again, improving again the MSA, improving again the distance contact. And this happens again and again and again a few times. They call this recycling until you get a much better output. Right. I'm going to stop here with the AlphaFold. I'll finish off with saying a few things about challenges of AI because, and this has nothing to do with biology only, but I think once you get into reading about AI and whatever, there's a lot of fear, and justifiably so. There's a lot of uncertainty. And I want to highlight some of the challenges. So it doesn't look like we don't care about these things just because we're trying to solve biology problems. So my number one challenge is, how does AI benefit everyone, yeah? And not just a handful of countries, not just a handful of people who are going to become rich because of it. So that's really essential, I think. I don't have answers to any of this, by the way. I'm just putting the questions forward. Okay? And each one of them is like a talk on its own. How do we deal with the environmental and societal impact? There's a huge environmental impact on training these models. If you have a model that has 300 trillion whatever connections, you're going to spend a lot of energy training it. Okay. And that energy has to come from somewhere. And there's a huge social impact. We know AI is going to threaten a lot of jobs. How do I align the AI models to human interests? How do I make sure that we are in control and that not they? I did say we might come to AGI one day. When we do, it will be too late. We have to find out now how to stop them from not interfering with our own interests. And how do we avoid biases in the models? I mean, that has become rather obvious very early in the history of AI. The models are biased because they read stuff off the web, and the web is biased. So we don't want this to propagate in the training data. Right. Now, that's my last slide. I'm sorry if I overrun. Anybody who is born, not that many people were born before the 80s here, but if you were early 80s or before, you may recognize who this guy is. This guy, Kasparov, and he's looking rather somber because he's losing the match here in 1997 to IBM's Deep Blue, which was the first computer to beat a grandmaster in chess. When this happened, there was this massive thing in the newspapers about how AI beat human intelligence and how we are inferior now to AI and so on. And I remember more specifically, actually, reading an article that has really changed the way I see things about AI. And that article had mentioned at the time, is AI better than human intelligence? And what the people put forward was that actually, you know what? Deep Blue was the collective effort of a lot of people. So Garry Kasparov did not get beaten by a machine. He got beaten by the collective effort of a lot of humans. Yeah. And I like this thought. I like this thought because it's sort of, I think it leaves you with an optimistic note that I think AI is like a cherished, a wonderful, a talented child. And the way you feel good about your child doing better in life than you, right? I think we should feel good about AI. It's our child. It's the child of collective human intelligence and should be proud of it. On that, I stop and thank you for your attention. Did I terribly overrun? Right.
Thank you very much for that, Irilenia. Before you leave, there might be some questions.
Oh, I thought you said the questions at the end.
After each presentation, sorry. Do we have any questions for Irilenia?
It was all so well explained, right?
I'm just going to check in case anyone has emailed in a question. But while I check, can I ask, it's actually about the energy consumption. If it takes so much energy to run these networks, why doesn't it take so much energy to run us?
We are extremely more efficient. I mean, we don't know why we are so efficient, but we are a lot more efficient than the machines. And that's a problem. But what DeepMind is trying to put forward is they're saying they're working on the problem of energy efficiency using AI. So they think AI will solve the energy efficiency problem for them. And they said they already cut down about 30% of the energy consumption they expected to have using AI. So they used AI to improve their energy consumption. And they are hoping, I mean, a lot of these people are really, I wouldn't say they're deluded, they're really very optimistic. And they're seeing the good of humanity and they're seeing these things actually helping us. And I want to believe in them. Like, I really do. It's just that, yeah, I don't always. Yes.
Thank you. Are there any questions from the floor? In that case, can we thank Irilenia again?